Good evening. So today is uh, good morning. Good evening. So today is a problem solving session for week 6, and the uh, lectures we will be covering for this session are lecture 1 and 2. They covers the basics of Talon part A and part B. Lecture 3 and 4, which includes design of Talon of genome editing part A and part B, and lecture 5 and 6, this includes application of Talon part A and part B. This course, genome editing and engineering, is being taken by um, so this course is being taken by Professor Utpal Bora. He is professor at Department of Biosciences and Bioengineering at IIT Guwahati. So I'll briefly introduce myself as well. I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in molecular virology lab at Department of Biosciences and Bioengineering at IIT Bombay Tech. Like in the previous session, so we will be having uh, questions. So we will go through each question, and we will also look for the concept for the each question, so that it can help you to solve your assignments. So let's begin with our question number 1. Question number 1 states, which system is responsible for secreting tail protein into plant cells? Option A is uh, T1 secretion system. Option B is T2 secretion system. Option C is T3 secretion system. Option D is T6 secretion system. The correct answer for this question is option C, T3 secretion system. So we will look into the concept. So tail, it stands for transcription activator like effector proteins, which are DNA binding proteins that are secreted by plant pathogenic bacteria such as Xanthomonas through the type 3 secretion system directly into the plant cell, where they act as a transcription factor to manipulate host gene expression, which favors their subsequent infection. So this this TAL protein is a bacterial protein which helps to infect its plant host. So this is secreted by one of the bacteria, which is Xanthomonas, which infects the rice plant. So once it infects the rice plant, so this TAL protein, they directly go into the nucleus of the plant cell, where it goes and bind to the promoter sequence, and which leads to the uptake, which leads to the control of their transcription and translation machinery. So the function of this uh, this TAL protein in the plant cell is it enters the plant cell nucleus, it binds to the specific promoter region of plant gene, it activates transcription of those genes, often leading to increased sugar up transport. It suppresses the immunity of the plant or other conditions which are favorable for the bacterial infection. So this is secreted by the type 3 secretion system. There are other secretion system in the bacteria, which are T1 secretion system, which controls the adhesion of the host to the host cell. So also the T2 secretion system, which controls the host cell degradation. They secrete such enzyme which leads to the host cell wall degradation. T3 the secretion system, the host cell manipulation, as we have seen here as well. So they go and bind to the promoter region and then control the transcription of a particular gene. So this is host cell manipulation and phytopathogenesis. So these conditions, they favors the bacterial infection. T4 secretion system, it helps in the bacterial competition and bacterial conjugation. T5 secretion system, it mediate the adhesion to the host cell or autoaggregation or biofilm formation. And T6 secretion system includes microbial competition. So uh, this TAL protein, which goes into the plant nucleus and take over the transcription machinery in order to increase its infection, is secreted by the T3 secretion system of the bacteria. So the correct option is option C, T3 secretion system. Let's move to our next question. Question second. Which gene is primarily targeted by PTH-XO1 TAL protein in rice? So the options are given on the screen. We need to answer which gene is responsible or the primarily targeted by PTH-XO1 TAL protein. So this is one of the TAL protein which is secreted by Xanthomonas in the rice plant cell nucleus. So this particular TAL protein, it goes and regulate or target which specific gene of the rice plant. correct answer for this question is option A, OS sweet 11. Let's see. So the structure of this PTHXO1 TAL protein is such that it has C terminus nuclear localization signal, which leads to the which help in importing of this protein to the rice cell nucleus. So, and downstream of this nuclear localization signal, there is acidic activation domain, which function as the transcriptional activator. Then we have a specific residue variable in two adjacent position determine the DNA binding in the host gene. So these are so so this region is made up of many repeats, and each repeats has around 30 to 35 amino acid sequence, and there are two variable sequence at the position of 12th and 13th, which determines the binding of this region to a specific base. So this is the DNA binding domain of the TAL protein. So this PTHXO1 directly activates the rice sugar transport gene, which is OS sweet 11. So this increases the sugar efflux in the apoplast, which provides nutrient to the pathogen and promoting disease or promoting the further infection. So the other TAL from zoo target different sweet gene. So other genes are OS sweet 13 and OS sweet 14, but this PTHXO1 it is specific for OS sweet 11. So how this functions, or the mechanism behind this, the takeover of this or the regulation of the transcription machinery of this OS sweet 11 gene is such that, so the bacterial pathogen, this Xanthomonas, it infects the rice leaf using its, as we have already seen in the previous question, using its type 3 secretion system, this uh, Xanthomonas injects its PTHXO1 TAL protein directly into the rice cell. Then, due to this nuclear localization signal present in the C terminus, this direct this PTHXO1 TAL protein into the plant cell nucleus. Then it has this DNA binding domain, or they have tail central repeat domain, which each domain consists of 33 to 35 amino acid residue. So this each repeats recognize one base pair of in the target DNA using this RVD repeat variable diresidue to specify which nucleotide is bound. So in this case, this PTHXO1 specifically recognizes a sequence in the promoter of the rice gene OS sweet 11. So after this recognition, activation of the host gene transcription occur. So this PTHXO1 has a transcriptional activation domain at the C terminus, or the downstream of this nuclear localization signal. So once it bounds to the OS sweet 11 promoter, this activation domain recruits the host transcriptional machinery. This results in the strong regulation, upregulation of the OS sweet 11 gene. And what are the physiological effect of this is, this OS sweet 11 encodes a sugar transporter in rice. Its overexpression increase sugar efflux into the apoplast, which is the space outside plant cell within leaf tissue. So these sugar act as a nutrient source for Xanthomonas, promoting bacterial multiplication and disease. So So this is how, so this sugar acts as a nutrient resource. So this is how this PTHXO1 tal protein is upregulating the sweet 11 gene, the upregulating the expression of the sugar efflux, which leads to the increase of sugar into the apoplast region. And this the bacteria which are present in this region, they tends to multiply in number and they tends to increase in their number. So how do the plant defend itself from this sort of um, this bacterial infection strategy? So some resistant rice varieties, they have a mutation in this OS sweet 11 promoter, which is this EBE, effector binding element. So what they do is they mutate or they remove this promoter region so that this PTHXO1 they cannot bind to it, and it leads to the resistance to the bacterial blight. So this is how this PTHXO1 um, is recognizing the promoter for OS sweet 11 gene, and it is uh, it this OS sweet 11 gene is targeted by the PTHXO tail protein in a rice plant. So the correct answer for this question is option A, OS sweet 11. So let's move to our next question, question 3. What is the primary function of talents in genome editing? Option A is protein degradation. Option B is gene expression inhibition. Option C is DNA cleavage at specific site. Option D is RNA interference. So we are at question number 3 right now, which states what is the primary function of talents in genome editing. So option A is protein degradation, option B is gene expression inhibition, option C is DNA cleavage at specific sites, and option D is RNA interference. So you can try to give the answer for this question. Then we will look into the concept of the question. So if you can try to answer this question. So the correct option for this question is option C, DNA cleavage at specific site. So let's see into the concept for this question. So the TALEN, it stands for transcription activator like effector nuclease. So this has two major parts. One is a DNA binding domain, so which are the TAL repeat. So this DNA binding domain, the TAL repeat or the TAL protein, as we have already seen, they are derived from Xanthomonas TAL proteins. They are made of around 33 to 35 amino acid repeats, and the each repeat is recognizing one DNA base pair. And this recognition, DNA base pair recognition is determined by this RVD, which is repeat variable diresidue at 12th and the 13th position in each repeats. So, so the amino acid which are present at the 12th and the 13th position, they determine the DNA specificity. While the entire sequence, it determines one DNA base pair, and this specificity is determined by the amino acid present at the 12th and the 13th position. So here are some of the amino acid which are present at 12th and the 13th position and recognizing the specific DNA base pair. So by arranging these repeats in series, the TALEN can program to bind any desired DNA sequence. So this is the structure of the TAL protein. In this question, it was asked what is the function of the TALEN. So in the previous lecture, previous session, we have studied about the FOC1 endonuclease. So the FOC1 is an endonuclease which is DNA cutting enzyme. It is fused with the TAL protein. So like during the zinc finger nucleases, we had attached this FOC1 endonuclease to the zinc finger proteins. Like this, these FOC1 endonuclease is also fused with the TAL proteins. So upon dimerization, so that each TAL protein is fused with the FOC1, and when these FOC1 they dimerized, uh, so this FOC1 they act as a dimer. So these two talons needs the two talons binding on the opposite DNA strand separated by the short spacer, which is usually 12 base pair to 20 base pair. So the FOC1 goes and cut or the cleave the double stranded DNA on this spacer. So when both the talons bind, the FOC1 domain come together and cut the DNA, creating a double strand break. And what are the additional feature of this TALEN? The nuclear localization signal, as we have seen that it has at the C terminus, it has nuclear localization signal domain, which helps in entering the TALEN into the nucleus. And the linker sequence, it connects the TALEN DNA binding with the FOC1 nuclease domain. So the correct option for this question is option C, DNA cleavage at specific site. I hope this question is clear. So let's move to our next question. Question number 4. Which enzyme is most commonly fused with tals to create talens? A is EcoR1, B FOC1, C BAMH1, D HIND3. We have already seen this in the previous question. So the correct option for this question is option B, FOC1. So the transcription talents, which states which talents which are transcription activator like effector nucleases are engineered by fusing TAL DNA binding domain, which is around 33 to 35 amino acid repeats, and the specificity of these TAL protein depends on the amino acid present at the 12th and the 13th position. So this each repeat they go and bind to one DNA base pair. So to make it a talon uh, protein, it is fused with an endonuclease, which is FOC1 endonuclease. So this FOC1 is a type 2S restriction enzyme, and this cuts the DNA at a defined distance from the recognition site. So when fused to TAL, the DNA binding function is provided by the TAL repeats, while the FOC1 only serves as a scissor to cut the DNA. So, so do so to FOC1 to function as an endonuclease, it has to be dimerized. So the two talons must bind the opposite DNA strand near each other to allow the cleavage. So to make a functional talon protein, so this TAL protein they are fused with the FOC1 endonuclease, and upon binding to the DNA sequence, so the one domain goes and binds to the upstream DNA sequence, while the other one goes and bound to the downstream DNA sequence, and once they get dimerized, the FOC1 they cleaves they cleave at the spacer sequence. So this is how the talons are created. So the correct answer for this question is option B, FOC1. let's move to our next question. Question 5. Question 5 states, which model organism was used for generating skid models via talent mediated knockout of PRKDC? So option A is mice, option B is zebrafish, option C is rats, and option D is goats. Please try to answer this question. So this question states, which model organism was used for generating skid models via talent mediated knockout of PRKDC. So you can try to answer this question. Then we will look into the concept of this question. You can try to answer this question this so that we can have an interactive session. The correct answer for this question is option B, zebrafish. Let's look into the concept. So the talon was among the first programmable nuclease used in zebrafish to generate gene knockouts. So in 2011, the researchers used talent mediated knockout of PRKDC in zebrafish to create a SCID, which stands for Severe Combined Immunodeficiency Model. So this PRKDC encodes for DNA dependent protein kinase catalytic subunit, which is essential for DNA double strand break repair during VDJ recombination in developing lymphocyte. So while the knocking out of this gene, it leads to the loss of functional T cell and B cell, and this zebrafish, it served as the immunodeficient zebrafish model, which is analogous to the SCIDs in humans. So to further study, so it can relate to the human by for studying the immune system development or for drug screening. So how the talents were designed for the knockout of this PRKDC gene in zebrafish. is such that the researcher the researcher, they first identified the target site in the PRKD gene, which in the exon 3. Here this is the PRKDC gene, and in the exon 3 they determine this uh, exon 3 as the target site. Then the two talons were designed. One is the left talon, which binds the upstream target site, and the right talon, which uh, uh, identifies the downstream, or on the opposite strand. Because the talons, to be functional, it has to be dimerized. So this fork1, they needs to be dimerized to function as endonuclease. So the two talons were designed, one which can go and bind to the upstream target site, and the other one which goes and binds to the downstream target site on the DNA. So this each talon contains the tal repeats, which as I have already told, the DNA binding specificity of the tal domain is responsible by the uh, amino acid which are present on the 12th and the 3rd position of these tal protein, and this tal repeat is fused with the fork1 nuclease enzyme. So this creates the entire talon uh, complex. So now, after the formation of the talon protein, they were delivered in the zebrafish embryos. So the talon encoding mRNA is synthesized in vitro, and this mRNA is micro injected into the single cell zebrafish embryos. So these were micro injected into the zebrafish embryo. So these embryo translate the mRNA, produce the functional talon proteins. So after the functional talon proteins are produced, the DNA binding and the cleavage has to happen. So these talon, they binds to their respective target site, flanking the spacer sequence. This is the spacer sequence which is covered by the fork1 endonuclease in the PRKDC gene. So this fork1 domain, they gets dimerized and introduce the double stranded break at the target site. So after this double stranded break, it has to go into the DNA repair pathway. So it gets repaired by the non-homologous end joining repair. Since we want this gene to be non-functional, so we do not provide any template donor DNA, so that it goes into the non-homologous end joining and create a frame shift mutation. So this frame shift mutation will lead to the loss of function of this particular gene, so that we can generate, or so that they could generate the skid zebrafish model. So this non-homologous end joining repair often introduce small insertion or deletion, which are the indels at the cut site. These indel can shift the reading frame, leads to the frame shift mutation, or they can also create premature stop codon, which will result in the truncative or non-functional PRKDC protein. So, so this protein is not functional in the zebrafish now, and by this method we can get, or the researcher could get a zebrafish which had knockout for PRKDC gene, which leads to the development of the skid zebrafish model. So what are the results for this knockout? So this embryo with biallelic mutation in PRKDC lack functional DNA dependent protein kinase activity. So this prevent the proper VDJ recombination, leading to the loss of functional T and B lymphocyte. So this has the skid phenotype, which is severe combined immunodeficiency phenotype in zebrafish, and these zebrafish, they can further be used as immunodeficient model for studying immune system development and human disease research. So this is how they created a zebrafish model to study the to study the immune system development related to humans. So this is how the talents are used for genetic engineering purpose as well. So the correct option for this question is option B, zebrafish. I hope this question is clear. I hope this question is clear. So let's move to our next question. Question number 6. Question number 6 states, which Talon assembly method uses hairpin capping oligonucleotides to improve assembly efficiency? First option is flash assembly. Option B is iterative capped assembly. C option is Gibson assembly. D option is star protocol. Please go through the question and try to answer this question. So the correct option for this question is iterative capped assembly. So let's look into the concept. So in this question, what is asked, which Talon assembly method uses hairpin capping oligonucleotide to improve assembly efficiency? So as it is already mentioned that these TEL effector proteins, they have these repetitive domains, and each domain has around 33 to 35 amino acid sequence, and the amino acid which are present at the 12th and the 13th position of this repeat is responsible for the binding to a one DNA base pair. So there are certain mechanisms by which these repeats are assembled. So one, so these are the, So this flash assembly, iterative capped assembly, Gibson assembly, and the star protocol, they are the different type of the Talon assembly method. So in iterative capped assembly, it uses the hairpin capping oligonucleotides to improve the assembly efficiency. So we will look into the concept how this method works for the Talon assembly. So here, so in the iterative capped assembly, it is a rapid, uh, a solid surface. This is the solid surface based DNA assembly method for creating the repeat modular uh, DNA like talons. So we have to make this repeats. So here you can see the final product is these A. It codes for A B C. It creates one talon repeat. Then we'll have again A B C. We'll have again A B C. So this is how we need to generate the talon repeats. So in this iterative capped assembly, which involves the sequentially adding individual DNA repeat monomer to a growing chain using capping oligonucleotides to block incomplete extension. So this technique prevents the accumulation. So this technique prevents the accumulation of the truncated product, significantly increasing the frequency of so this technique prevents the accumulation of truncated products, significantly increasing the frequency of full-length final construct and allowing for the efficient, scalable synthesis of complex. So the how iterative capped assembly works. So we have first solid phase immobilization. So this process begins by immobilizing a DNA template, often a biotinylated initiator oligonucleotide onto a streptavidin beads. So then we add the monomer addition step. The first DNA monomer, which is A here, is ligated to the initiator. So then capping and washing. This is the capping oligonucleotide is then added, which is designed to be compatible with the extension of any partially ligated, incompatible with the extension of any partially ligated chain. So this capping oligonucleotide block the 3' end of any incomplete product, preventing it from further growing. So the beads are often washed. So these beads are often washed to remove the excess reagent and any unligated capping oligos. So this cycle is then again repeated. So this process is repeated in a cyclic fashion using a different monomer B, A, B, C in a sequential ligation wash pattern. So a key feature is that the next monomer is in the sequence, example B after A, is ligated to the end of the successfully extended chain. So then the termination occurs. So when the desired length is reached, a terminator oligonucleotide is added instead of a regular monomer to finalize the construct. So then the final elution occurs. Then this has to go and inserted in or cloned into a compatible vector. So this can be used for the genome editing purpose. While in case of flash assembly, it uses a high throughput ligation approach, but it does not involve hairpin capping. In the Gibson assembly, the general DNA assembly method using exonuclease, polymerase and ligase, not specific to TAL or hairpin caps. While in the STAR protocol, a different TAL assembly method rely on the sequential ligation without the hairpin capping. So the correct option for this question is option B, iterative capped assembly. So these repeats has to be ligated to form a functional domain. So there are different methods where how these repetitive sequence are assembled. So you can read on this more elaborately. So there are how each method works in order to assemble these talon or the tail effector proteins to become a functional talon protein. So the correct option for this question is option B, iterative capped assembly. So if this question is clear, let's move to our next question. Question 7. Question 7 state, what is a common outcome of talons targeting mitochondrial DNA? Option A is HDR based recombination. Option B is NHEJ mediated repair. Option C is rapid degradation of mitochondrial DNA. Option D is activation of nuclear encoded mitochondrial genes. Please go through the option and try to answer this question. So you can try to answer this question so that we can have an interactive session.
Rapid degradation of mitochondrial DNA.
Yeah, the correct option is option C, rapid degradation of mitochondrial DNA. So let's look into the concept. So let's look into the first the structure of the mitotalons. So these mitochondrial targeting sequences. So these mitotalons, they have a specific domain or a specific sequence which is attached at the end terminus of these tal protein. This is mitochondrial targeting sequence. So because of these specific sequence, so these talons, they can be used uh, uh, they can be used to insert into the mitochondria, and they can go and bind to this mitochondrial genome. So they are usually short peptides made of around 20 to 40 amino acid sequence. So they are derived from the naturally mitochondrial localized protein, which example cytochrome C oxidase subunits. So this ensure the talon protein to be imported into the mitochondrial matrix where the mitochondrial DNA resides. So like the previous structure, these mitotalons, they will be having the tail DNA binding domain, which is the core of the talon structure. They are again they are composed of around 33 to 35 amino acid repeats, and each repeats recognize the one specific DNA base. Uh, so at the 12th and the 13th position, they have repeat variable di residue. So based on the sequence of these amino acid present at the 12th and the 13th position, they recognize one specific DNA base pair. They are arranged in tandem to specifically bind and chosen the mutant mitochondrial DNA sequence. Then this is attached to the fork1 endonuclease. So this is attached or the fused at the C terminal of the TAL domain. Again, it has to be dimerized to properly function. So this we need the two functional talons, so that the one talon will go and bind to the upstream region, and one talon and go and bind to the downstream DNA sequence. So once this fork1 endonuclease they gets so first, once these DNA binding domain goes and recognize the sequence on the mitochondrial DNA, so these fork1 they gets dimerized and cut on the double strand DNA sequence, leading to the double stranded break. So these talon they can be engineered with mitochondrial targeting sequence to localize them into the mitochondria. So they are called the mitotalons. So how we have seen how they are engineered. So in the mitochondria, what happens is there is no DNA repair mechanism like homologous directed repair or the non-homologous end joining. Therefore, the mitochondria cannot repair double-stranded break. So as a result, cut mitochondrial DNA is degraded instead of being repaired. So if there is a mitochondrial DNA which is mutant, so what researchers do is they engineer a talon which can go and specifically bind to the mutated mitochondrial DNA, and this will lead to the double stranded break in the mutated mitochondrial DNA, and these break will not be repaired because the mitochondria does not have the homologous direct repair or non-homologous end joining repair. So if they are not repaired after the double stranded break, they will get degraded, and only we will be left with the wild type mitochondrial DNA. So broken mitochondrial DNA is recognized as damage. So the mitochondria uses nuclease and exonuclease to degrade the cut DNA fragments. This prevents the accumulation of defective mitochondrial DNA. So human cell usually contains a mixture of wild type and a mutant mitochondrial DNA. So after mitotalon treatment, the mutant mitochondrial DNA is selectively degraded, while the wild type mitochondrial DNA they would remain intact. So over time, we'll having the large population of the wild type mitochondrial DNA as compared to the mutated uh, mitochondrial DNA. So what will be the outcome? The reduction or elimination of mutant mitochondrial DNA load, restoring the normal mitochondrial function. So the correct option for this question is option C, rapid degradation of mitochondrial DNA. So I hope this question is clear. if this question is clear, let's move to our next question. Question number 8. Question number 8 states, which gene was targeted in talent-based knock-in experiments in rats for studying the glucocorticoid receptor functionality? So you can go through the option and try to answer this question. You can try to answer this question. We will look into the concept. So the answer can be wrong. It's no problem. You can try to answer.
NR3C1 gene.
Yeah, the correct option is NR3C1 gene. So let's look into the concept. So this NR3C1, it encodes for the glucocorticoid receptor in rat. So this talent-based knock-in experiment in rats, they targeted NR3C1 to insert a reporter sequence or a mutation. So why did they target this NR3C1 gene? So this codes, as it is mentioned, that it codes for the glucocorticoid receptor. So this glucocorticoid receptor, it involves in the various pathways or various function, uh, which is glucocorticoid receptor signaling, stress response, hormonal regulation, or the protein synthesis degradation mechanism. So glucose uptake, etc. So after which the the knock-in of this gene, the knock-in into this gene, or the mutating by after mutating this gene, so they can study the effect of the glucocorticoid receptor signaling or the stress response or the hormonal regulation in vivo, and then they can also relate it to the human. So how, so the again, the process is similar as we have seen in the mitochondrial DNA as well. So first the target region has to be determined. So this is the gene for NR3C1 gene, and at the exon 3 the target site was decided where they mutated this G. So once they have decided the targeted site, so they chose a gene of interest in a rat C6 glioma cell. So they identified the 20 to 30 base pair target site within the coding or the regulatory region. So this could be either regulatory region or it could be a protein or the coding region. So they made sure that the target sequence it starts from 5 prime T. So this is the requirement for the tail binding. Then the talon were designed. The two talons were designed to bind to the opposite strand with a spacer of uh, 12 to 20 base pair in between, so that the fork1 endonuclease gets attached to it and introduce the double stranded break. So further, after the formation of the talon, so this talon was delivered. Talon sequence are cloned into the expression plasmid under a strong promoter, and the these cells are transfected either via liposuction or electroporation. Now we have introduced a gene in a mouse which uh, which is the mutated version of this NR3C1 gene, and then we can study the phenotype of the rat via this mutant gene, will give which will allow us to study the glucocorticoid receptor signal in other stress response or hormonal regulation or other function based on the function of these glucocorticoid receptors. So this is how the talons are either used for the gene knockout or a gene knock-in studies. So the experiment can be designed if you want to study the function of a particular gene. So what is the effect of a particular gene in a particular phenotype? Then you have to either introduce a particular gene or you have to either delete a particular gene. So these talent strategies, they can be used for this genome editing. So these phenotypes can be studied. So the correct option for this question is NR3C1. So the basic idea here is, first you need to make the TAL effector protein based on your target sequence. First you need to determine the target sequence and then make a TAL effector protein. Once you have your TAL effector protein, you fuse this TAL protein with fork1 endonuclease. So and then you clone this entire segment into a plasmid, and this plasmid then transfected into a particular cell type. So once they express this talon, it goes and binds to a particular DNA sequence that we have already chosen for its target site, and this fork1 which cleaves the particular site. So after that, based on either homologous recombination or the non-homologous end joining repair pathway, it will create a mutation or it will knock out a gene or knock in a gene. So based on your objective of study, you can either knock out a gene or knock in a gene. So this depends on your objective. So basically this is how the talents are synthesized and are used for genome editing purpose. So I hope this question is clear. So let's move to our next question, question 9. So question 9 states, which mechanism is used to achieve high targeting specificity with TAL based editor? A option is splitting the deaminase into two non-toxic halves. B option is using full length DA domains. C option is co-delivery of talon and RNA components. D option is incorporating a fluorescent protein. Please try to answer this question. Please try to answer this question. So the correct option for this question is option A, splitting the deaminase into two non-toxic half. So let's look into the concept first. So the split DTA tail base editor system is primarily applied in a mitochondrial genome. So unlike a nuclear genome, this mitochondrial DNA, it lacks efficient repair pathway like homologous direct repair pathway or non-homologous end joining. So these tail protein can be fused to the mitochondrial targeting sequence, allowing a specific binding to mitochondrial DNA. I think my screen is not clearly visible, so just let me fix this first. Still not clear. I think it will remain the same. So I'll just explain like this only so that you can also understand. So this tail base editor, they are derived from mitotails. They are fused to bacterial cytidine deaminase. So why the they are fused with bacterial cytidine deaminase? Because these bacterial cytidine deaminase, they uh, so they can introduce mutation, so they can convert the CG to TA. So so there are uh, uh, there are already mutation which are present in the mitochondrial DNA. So to correct these mutations, so these bacterial cytidine deaminase, they are added or they are fused with the tal protein, so they can go and introduce they can replace the particular mutation, so they can convert the CG into the TA and then can correct the mitochondrial DNA. So but if these these are attached at basically they are attached at the C terminus, but when they are attached at the C terminus, what they do is they go and bind to the non-specific mitochondrial target sequence. They can go and attach to anywhere and can base edit. So this is not feasible. They can go and further mutate the mitochondrial DNA. So the what scientists has done, they have divided this DDA into the two fragments. They joined one fragment at the N terminus and joined one fragment at the C terminus. And after splitting these DDA fragments, they could achieve the specific binding of these mitotalins to the mitochondrial DNA. So each half is fused to the tail binding domain that binds to the adjacent site on the opposite DNA strand. And only when both the tails bind next to each other does these DA half reconstitute into an active enzyme, leading to the site specific CG to TA editing in the double stranded DNA. So this split strategy greatly reduces off target editing and it ensures the high targeting specificity. So that is why these DDA are split, and one DDA goes and bind to the one TAL protein, which goes and bind to the upstream DNA site, and one goes and bind to the TAL protein which binds to the downstream DNA sequence. And only when they are properly go and bind to these target sites, these DDA become functional, and then they can they lead to these site specific CG to TA editing. So this is how they are increasing the targeting specificity. So the correct option for this question is splitting the deaminase into two non-toxic halves. So I hope this question is clear. So let's move to our next question, question number 10. Question number 10 states, what is the main advantage of using TALEN over ZFN or zinc finger nucleases for genome editing? A option is smaller cDNA size, B option is lower cost and ease of delivery, C option is simpler protein DNA recognition code, D option is higher specificity and lower off target effects. So the correct option is already given on the screen. So it is the correct option is simpler protein DNA recognition code. So in zinc finger nuclease, as we have already studied in the previous lectures, it uses the zinc finger domain to recognize the 3 base pair DNA sequence. So designing a new zinc finger for a specific DNA target, it can be complicated because each zinc finger binding domain can be influenced by its neighbor, making the code context dependent. So we have already seen that how these one zinc finger binding domain is affected by its neighboring domain. So we will not go to this here. So this is make this this zinc finger nuclease more complicated than that of the TALEN. In case of a TALEN, each TALEN repeat it goes and uh, bind to the specific one DNA base pair, and this is not affected by its so these TALENs on the other hand uses the TAL repeats, where the each repeat specifically recognize a single DNA base. This makes designing TALENs much more straightforward and modular compared to ZFN. So other options were not correct. The smaller DNA size, the TALENs are actually larger than ZFN, so it is false. So the lower cost and ease of delivery. So since the TALENs are much larger and they become hard to deliver than that of the zinc finger nuclease, so this is also false. The option is higher specificity and lower off target effects. So while TALENs they can be very specific, the main advantage over ZFN is the simpler design, not necessarily the off target profile. So there are some differences between the zinc finger nucleases and the talons. So in zinc finger, zinc finger motif, zinc finger motif, they recognize the 3 base pair, while the each talon repeat it recognize 1 DNA base pair. The recognition is context dependent in case of the zinc finger nuclease. So the neighboring zinc finger domain, it affects the other binding capability of a zinc finger domain. While in case of talon, it is simpler and modular, one repeat per base, so it is very predictable. Ease of design for zinc finger, it is complex and challenging. For talon, it is relatively simpler and straightforward. Size for zinc talon, the size is much larger than that of the zinc finger nuclease. So the delivery for zinc finger nuclease is comparatively easier because it is smaller in size, while it is harder because of the larger in size in case of talons. Specificity, uh, in zinc finger nuclease, off targets are quite possible, while in talons, off targets are less possible. So there are other few differences. So this is how the differences are there. So the correct option for this question is option C, simpler protein recognition domain. So this was the last question. So we can go through the first 5 question again. So you can also see what were the rest of the question, because I think you joined in late. So the first question was, which system is responsible for secreting TAL protein into the plant cell? So the correct option for this question was option C, T3 secretion system. So the TAL, so as it is already mentioned, the TAL stands for transcription activator like effector proteins. They are the DNA binding proteins and are secreted by plant pathogenic bacteria, Xanthomonas, and they are secreted by the type 3 secretion system of the bacteria into the plant cell directly, and which further regulates the transcription machinery of the plant, which will help its further bacterial infection. So there are other secretion system in the bacteria. T1 secretion system, it regulates the adhesion to host cell, while the T2 secretion system, it regulates the host cell wall degradation. T3 secretion system, it regulates the host cell manipulation and phytopathogenesis by secreting the TAL protein into the nucleus of the plant cell, and which overtakes the transcription machinery in order to increase its infection. T4 secretion system, it regulates the bacterial competition and bacterial conjugation. T5 secretion system, it regulates adhesion to host cell, auto aggregation and biofilm formation. T6 secretion system, it involves in the microbial competition. The correct option for the question 1 was T3 secretion system. Option 2 was, which gene is primarily targeted by PTH-XO1 TAL protein in rice? You can try to answer this question. So the correct option is option A, OS sweet 11. So the structure of this PTH-XO1 TAL protein is such that it has on C terminus, it has this nuclear localization signal, which helps in importing of this protein into the nucleus of the rice plant, and it downstream of this, it has acidic activation domain, which function as the transcriptional activator. So after going into the nucleus of the rice plant, it goes and bind to the promoter of OS sweet 11 gene, and it codes for and this and activates the transcription of this gene. So after the transcription, it leads to the synthesis of the uh, it leads to the synthesis synthesis of sugar transporter in rice, which leads to the increase of sugar in the apoplast space apoplast of the plant cell, where these bacteria resides. So after the sugar gets into the apoplast space, they serves as the food for these bacteria. So this will lead to the increase in the bacterial number, and it will help in further bacterial infection. So this is how they go and bind to the promoter of this gene and regulate the transcription of the sugar transporter, which will lead the increase of sugar in the apoplastic region. So what plant does in their defense mechanism, they delete this promoter site of this specific gene, so that these TAL protein, they don't go and bind to this specific promoter, and further the transcription also stops. So the correct option for this question is option A, OS sweet 11. So the third question is, what is the primary function of talents in genome editing? We have already seen that uh, it results in the DNA cleavage at specific site. So talents, they are transcription activator like effector nuclease. They have two major parts. One is the TAL repeats, the other and fuse with the FOC1 nuclease. So TAL repeats are designed in such a way that it goes and recognizes the DNA target site. So they have around 33 to 35 amino acid repeat sequence, and they differ in 12th and 13th base pair sequence. So this is how they are designed. And these FOC1 endonuclease, they go and cleave these target site. So the correct option is option C, DNA cleavage at specific site. I hope the third question is clear. So what is the primary function of the TALEN in genome editing? The correct option is DNA cleavage at specific site. So we have already seen how these talents are designed based on their target site. So these each TALEN repeats, they are composed of amino acid residue, which are around 33 to 35 amino acid residue long, and they differ in 12th and 13th position, and based on this difference, they go and bind to their target site. So these TAL is, they are further fused with endonuclease, which is FOC1 endonuclease. So once these entire structure is formed, once these DNA binding domain go and binds the DNA target site, these FOC1, they go and cleave the double stranded, they go and leads to the double stranded break onto the DNA. So further it will lead to either any of the DNA repair mechanism, either non-homologous end joining or homologous recombination pathway. So the primary function of TALEN is the DNA cleavage at specific site. Question fourth is, which enzyme is most commonly fused with TALs to create TALENs? That we have already seen. It is enzyme FOC1. It is an type 2 restriction enzyme that cuts the DNA at defined distance away from its recognition site. So these DNA binding domain, they recognize the target site, and what FOC1 does is it cleaves a defined distance away from the recognition site. So it cleaves at a certain position of the on the spacer sequence. So the FOC1 endonuclease, it is attached with TAL protein to make an effector TALEN protein. So the correct option is option B. Well, the question 5 states, which model organism was used for generating SCID models via TALEN mediated knockout of PRKDC? So you can try to answer this question.
Zebrafish.
Yeah, the correct option is option B, zebrafish. So, so the designing is the same that we have already seen in the previous question. So while this gene PRKDC, it so the knockout of this particular gene, it creates the SCID model of the zebrafish, which is severe combined immunodeficiency model. So this gene, it encodes for DNA dependent protein kinase catalytic subunit. It is essential for DNA double stranded break repair during the VDJ recombination in developing lymphocyte. So after knocking out of this gene, it will lead to the loss of functional T lymphocyte and the B lymphocyte, which will result in the immunodeficient zebrafish. So this zebrafish then further be used to study the immune system development and for the drug screening purpose. So that is why these gene knockouts are formed in zebrafish. So why in the zebrafish? Because it is useful vertebrate model to study the immune system development and for drug screening. It is also very cost effective. It is cost effective than the rat model, so that is why the zebrafish are also used. So the correct option is option B, zebrafish. So I think you joined from question 6. If you have any doubt in any question, you can ask.
Can you explain HSC to RTA conversion?
You are not audible clearly. Can you speak a little louder? Or can you write in the question in the chat box? Do you have any question? So if you don't have any question, you can leave the meeting because I'm not sure if you have any question. I could not hear you properly.
Ma'am, can you explain mechanism of CC to TA conversion?
Mechanism I have heard, but I can you write in the chat box? I think you are asking about the zinc finger nucleases, if I have heard correctly. The mechanism is so okay. So the mechanism is, so there is a bacterial protein which is cytidine deaminase. So what it does is it removes the amine group from the cytosine and it will result in the thymine formation. So this is how this enzyme is used for the conversion of this Cg to Ta. So after this C is converted to T, further the replication will occur, the two strands will get separated and this one strand will have the Ta and the other strand will have a Cg. So this is how the mutation will be expanded in the population. So this is a deaminase, so it removes the amine group from a particular base. I hope this is clear.
Yes, ma'am.
Okay, so do you have other questions?
No, ma'am.
Okay, all right. So if you don't have any other question, you can leave the meeting. So we will again have the class next Tuesday at the same time. So thank you for joining in.
Thank you, ma'am.